Welcome. This little video is going to serve as an introduction to the mechanics of the experiment we're going to be doing this week on bacterial transformation. We're going to be using a plasmid, which we'll learn about in a moment, which carries the gene for green fluorescent protein, which was actually isolated from a jellyfish, as you can see here on the screen. Before we go into the actual mechanics of the experiment, we need to review a quick central framework or dogma of molecular biology. So you know everything stems from DNA, which we know DNA ultimately leads to RNA, messenger RNA, when it gets transcribed, and then when translated it results in a protein or proteins, which ultimately will lead to the expression of what we refer to as a trait. So in keeping that in mind, we'll be able to keep track of sort of what's going on during this process of bacterial transformation. First things first, we need to get ourselves sort of situated. If this is a typical sketch of a bacterial cell areas, just to make sure we know what we're talking about, you have the cell wall on the outside, which will be important when we start talking about how the antibiotic resistance works and the antibiotic susceptibility. The pink squiggly line there represents the bacterial chromosome, which has all the genes that are necessary for the cell to survive. We're going to use plasmids, which are little circular pieces of DNA that are known as extrachromosomal DNA. In this case, we're using a plasmid that's known as P-Glow. It's proprietary from Biorad. It's called P-Glow because it has the gene for green fluorescent protein, which will make the bacteria glow if they're expressing the gene that's on that plasmid. The process of transformation actually involves getting the plasmid into the cell. So taking this plasmid here and getting it into the cell somehow. We're going to be doing what's known as artificial transformation. We're going to be forcing these plasmids into the cells. This does happen naturally. Cells exchange plasmids all the time. That's how bacterial antibiotic resistance can spread between cells. Once the plasmid is in there, if it gets expressed, it will produce the proteins that are coded for on that plasmid. And if it has another region that we'll see in a second, it will be able to be passed on to any future cells. This one's showing that we could produce two proteins on this of interest, the GFP, the green fluorescent protein. We're also going to need to have some kind of antibiotic marker or resistance that we can test for so that we can select for cells that have been transformed. And we're going to use what's the gene for beta-lactamase, or the BLA gene, which will confer ampicillin resistance to the bacteria if they are indeed transformed. So that's a quickie overview of what transformation is. We'll be doing that in the lab. We need to go in a little bit about how the plasmid works and all that in order for you to have a sense of what's going on this week. First of all, the plasmid itself. Here's a diagram of what the plasmid would look like. It has a number of areas that are of importance and interest to us. The first one is known as the ORI, or the origin of replication. If a plasmid is going to be of any use to a cell going forward, it needs to be able to replicate, just like when the chromosome replicates. So there is this region here, the ORI site, which is the origin of replication, where DNA polymerase would bind and make a copy of it before the cell divides. Another area of interest on here is the BLA gene, or the beta-lactamase gene. That gene codes for an enzyme ampicillinase, which would give the bacteria the ability to break down ampicillin and therefore be resistant to ampicillin. Two other genes of interest on here are the ARA, or the arabinose gene, which codes for a protein involved in the operon, which we'll see in a second, and then also the GFP gene, which is the gene for green fluorescent protein. Ultimately, we're going to be trying to get the bacteria that were not resistant to ampicillin to become resistant and be able to express this GFP gene. The way it works, there's something called an operon. An operon is a collection of genes in any of the regulatory sequences involved. So this entire area here that's circled is known as an operon. This happens to be the arabinose operon because it's involved in the metabolism of arabinose. The areas of interest here, there are three genes sort of downstream. If that thick dark line represents the DNA, those three areas code for three different genes involved in arabinose metabolism. They're shorthand arabinose B, A, and D for the three different gene products. There's also a region upstream of those coding genes, which is shown here underneath this arabinose C protein. That's the promoter sequence, which is where, if the gene is going to be expressed, RNA polymerase needs to be able to bind. The way it's being shown here, the arabinose C protein is bound on the promoter sequence and prevents the RNA polymerase from actually binding. So this gene would be turned off right now. An operon allows us to turn on and turn off genes. In this case, the molecule that will induce or turn the gene on is arabinose. Obviously, if arabinose is present, we'd like to be able to metabolize it, or the cell would like to be able to metabolize it. The way that works is the arabinose actually binds with the arabinose C protein and changes its shape. You see here it's in a different conformation than it was above. By changing its shape, it actually exposes the promoter region, which is where the RNA polymerase, which is the protein that is responsible for making messenger RNA, can now bind to the promoter sequence, since the arabinose C with the arabinose bound to it has changed its shape. The polymerase can bind and then go down the gene and actually transcribe the genes in this direction, left to right, and make the messenger RNA for the arabinose B, A, and D. This is the basic operation of our mechanics for how an operon works. In our case here, the people at Bio-Rad have engineered the plasmid to make it a little bit different. You see on the next page here. Here's what happens is they've taken the B, A, and D genes and removed them and replaced them with downstream. Here's the same arabinose C promoter area being bound to the arabinose C, but downstream of it we no longer have the three arabinose metabolizing proteins. Instead we have the gene for green fluorescent protein. So what happens is, because this operon is still controlled by that arabinose C area, when we grow the cells in the presence of arabinose, right, same molecule that will sort of turn on the gene, it will bind to the arabinose C protein and change its shape so that the polymerase can now bind. And instead of going downstream and making the three proteins involved in arabinose metabolism, instead the cell will be able to transcribe the gene for green fluorescent protein, which will then hopefully be expressed, translated into the actual GFP itself, which when we shine ultraviolet light on the bugs, they will glow green, just like that jellyfish back on the first slide. So that's the quick and dirty for how bacterial transformation works. Again, we're going to try to get this plasmid into the cells and then find the ones that are expressing both the antibiotic resistance, because they have the BLA gene, and ones that are hopefully expressing the green fluorescent protein gene. So that's what we're going to be doing this week in the lab. Thanks.